Welcome back, everybody. Hope you had a lovely lunch, and lunch also for people watching at home as well. So we're now going to move on to our second panel discussion of the day. Delighted to welcome Professor Gerry Wilson and Professor Ursula Fearon, our two chairs of rheumatology. So first of all, if I could just introduce yourself, Gerry. So Professor Gerry Wilson was first appointed as the Arthritis Ireland Chair of Rheumatology in UCD in 2013. Gerry is also a practicing consultant rheumatologist as well. And then we have Professor Ursula Fearon, who was appointed to the Chair of Rheumatology, Molecular Rheumatology in Trinity in 2015. So you're both very, very welcome. Delighted that you could join us here on our important 40th anniversary celebration. So Gerry, if I can maybe turn to you first. Look, it was a long-term goal of Arthritis Ireland to invest in infrastructure, in research in rheumatology, and to recognise the important role that research plays in, transforming the lives of people with arthritis. Can you tell us more about how the establishment of the chairs of rheumatology with Arthritis Ireland has assisted the progress of research regarding arthritis?
Yeah, well, thank you, and thanks to Arthritis Ireland for the support, actually, in developing it. And people have probably heard me saying, a lot of people have heard me saying, one of the great things about rheumatology in Ireland, and it has been published, that we're coming from a very strong base, and that was before the chairs were created. Ireland was actually at the top country in the world in terms of quality of research, published research. So there are many ways of judging the quality of research, and one of the best ways is how many times what you publish is quoted by your peers. Mm-hmm. And Ireland, actually, it was a survey of 35 richest countries in the world, and they looked at the metric of rheumatology publications in these 35 countries and how many times paper in rheumatology was quoted by peers across the world, and Ireland actually was way ahead of any country. The metric was 47 for any paper published between, I think it was 1995 and 2010. And the next country was the Netherlands, Holland, which is a great country for research, and it was 40. And then I think Belgium was 39. It was just amazing to see the excellence. So we were Arthritis Ireland, I think, has just really developed and built upon that excellence that was there from all of the researchers who were, both basic scientists and clinical. And there weren't any sort of clinical academic positions there, but it was clinicians. And one of the things I noticed coming from the NHS, so I was in Sheffield for a long time, is that there's much more sort of interest, I think, among virtually every consultant I would work with in doing research. So they've all been away and worked either in Britain or particularly in the States, and they've got a very good research training. They've generally done MDs or PhDs, and then they come back and they're keen to pursue their interest in rheumatology. And so most of the consultants across the country are very good at that. I was just down in Limerick yesterday, actually. I was giving a talk, and that was due to the fact that the clinicians down there had, discovered some families with particular form of inflammation, inflammatory disease called Behçet's, which, and they contacted me because they had an interest in research, and we've taken that forward and got, grants and some nice findings from that. So what I think the chairs do is builds on a very strong research background and research interest. And it's really important to build the infrastructure. And I think from Ursula and myself, the perspective is particularly we want to train the next generation of researchers and to try to help Arthritis Ireland pump prime key areas of research, areas that are not particularly researched. That would be great. There's so much interest and expertise across the country in lots of things, and we'll probably come on to the research prioritization exercise that Arthritis Ireland performed, looking at areas of research that people across the country want us to, or Arthritis Ireland, to take an interest in. But it's really, if we can pump prime those areas, that will further build strength of rheumatology research across the country.
Okay. And can you tell us maybe a little bit about, the recent developments in your area, in your area of research, since you became the chair of rheumatology, and obviously you can maybe tell the audience about Mashir and all of that, and how that might impact, upon patient outcomes in future years.
Yeah. So, so what my sort of key interest has always sort of been genetics and trying to understand. So genetics is important so it's in rheumatoid. So it's not just about who gets the condition, but it's who gets severe rheumatoid. So who gets the persistent type of rheumatoid, and who gets X-ray damage to the hands and feet. There's an allied condition which is a similar genetic background, type 1 diabetes. So that's autoimmune as well, where the immune system breaks down and you develop immunity against self-constituents. So in type 1 diabetes, patients present when the immune attack has destroyed all of the insulin-secreting beta cells. So by that stage, you've missed the boat. Whereas in rheumatoid, generally patients present with very little or no joint damage. And so one of the questions there is, how can you identify who's going to develop severe forms of rheumatoid? There are a few things that we do clinically, like if the antibodies are positive in the blood, or high level of inflammation in the blood. But we don't know, they're not highly predictive. So I've been using genetics to see if we can predict and find genes and proteins that make things worse. And we've been successful in identifying a few novel proteins or genes that minor variants in them are associated with more severe damage. And Arthritis Ireland funded some of the work on a particular gene that we discovered that way. And the other thing is using the new genetics. So the technology for genetics is just amazing compared to when I was an ARC fellow, arthritis research, and I did a PhD in Sheffield in the early 90s, and I spent 18 months. My first 18 months, I sequenced 1,000 bases of DNA in 10 people in the TNF gene looking for genetics. And now I send off samples of blood on patients, and I get it done commercially, where they sequence every one of the 21,000 genes, and they'll send back the results within 3 to 4 weeks. And then it's funny how medicine has changed, research has changed. You get this ton of data, and then it's a needle in a haystack, because we're all full of genetic variants. And if I do take your DNA and sequence your 21,000 genes, I'll find 40,000 variants from the reference human gene. And then we have to work out which of the one is the disease causing change.
A small job.
So it's like a lot of medicine. These days we generate so much data that certainly where I work, and I'm sure Ursula, the sort of buildings are full of mathematicians and people. A lot of them are engineers who come out, and they're great with data, and they're great with writing their programs to analyze and look for these needles in that big haystack of data.
Yeah. And actually that leads nicely on to my next question for Ursula, which is that, you work in the field of translational research. Can you tell us a little bit more about what that actually means? Yeah. And why, the interpretation of data in relation to patients is so important?
Yeah, that's really, really important. And actually the word translational research gets thrown around a bit. A lot of scientists and clinicians talk about doing translational research, but actually translational research, the term that used to be used for it is bench to bedside, but really it's a full circle. It should be bedside to bench to bedside. So everything we do, a lot of my work is based on trying to find, similar to Gerry, protein and genes that predict disease onset, predict response to treatment or predict response to a certain treatment, so you get the right treatment the very first time. But when we measure a protein in the lab and it's high, we have to kind of go back and see, well, what was different about that patient? Because we could measure a protein and we get this, some patients have really high levels of the protein, some patients have moderate levels, and some are the same as healthy controls. So a lot of what I do is trying to figure out what's different about the patients that have really high levels versus the patients that have really low levels. And how we try to interpret that is that we go back to the clinician and ask them, well, what was the clinical status of this patient? Had they very high disease activity when they came in to the clinic? And then what we do is we follow. So while we measure the proteins and genes on day one, sometimes we have to wait a year to actually interpret the data because we follow the patient to see whether the patient responded to their treatment. And what we do is, can we figure out with the protein that we measured the very first day you came into the clinic, can that predict whether you're going to respond to a treatment or you're not going to respond to a treatment? And then when we find out who responded and who didn't respond, we go back to the baseline data and say, well, is there something different in those proteins that will tell us on day one that that patient shouldn't be put on this drug because they're never going to respond to it? So actually, what we do in the lab is really important, and the patients are really important. I can't do anything in the lab without the patients. I get blood and I get tissue samples from patients, but I can't interpret any of the data that I get in the lab unless we have the patients involved and we have the clinicians involved. So it's a full circle. It's the patient coming in to meet the clinician. They go through their routine clinical assessments, and if they want to get involved in research, they consent to be involved in research, and then I get blood and tissue samples. But everything I do with that is then interpreted straight back to that patient. So that's what we mean by translational research. But a lot of where it stops is where the basic scientist or even the clinician measure a protein and gene and just say it's high in the patient, but actually don't go back to the individual patient. And it's coming back to Gerry's point that we're all individually different. So sometimes it is like searching for a needle in a haystack, but it's so important. And it comes to personalized medicine as well.
Yeah, and that's where my next question is going. In that, much of your research focuses on understanding, the underlying mechanisms that drive these diseases, and and what treatments will work. Ultimately, it's about what treatments will work for different patients.
Yeah.
Can you tell us just a little bit more about how research is helping us to understand how patients will respond to the treatments that are prescribed? Because we know that, look, it's been the last 20 years or 25 years, it's, particularly in relation to inflammatory arthritis, there's been a real revolution in terms of treatments available. But it's about that next step in terms of how...
So actually, if I go back, you've led me into, so I go back 20 years. So 20 years ago, 25 years ago, the treatments for patients, so I work on rheumatoid arthritis and psoriatic arthritis, and the treatments were pretty poor, and the prognosis was pretty poor. But then researchers in the Kennedy Institute in Oxford, well, they're in Oxford now, they're in London then, they started to take biopsies from patients, and that's the nearest you can get to actually putting a drug, rather than putting a drug into a patient, putting it into the tissue that you get, because that reflects the patient. So if you get a biopsy from a patient, it's the nearest you're going to get to what the information is inside in the patient. So they actually put this drug, an inhibitor against a molecule called anti-TNF, which I suspect a lot of you have either been on or are at the moment. And so they used the patient's tissue to identify that that protein could be blocked. And that basically revolutionized the whole, field for treatment of inflammatory arthritis. So I suppose I split what I do into three aspects. One aspect is that we look at patients that don't respond to any treatment. And so we do get biopsies from those patients, and we do an awful lot of work with industry, and we test an awful lot of inhibitors in the lab in these biopsies of patients that aren't responding, because we're trying to find new therapeutic targets. So that's one aspect of what we're doing. Then the second aspect, which are kind of the holy grails really, is trying to predict disease onset and trying to predict who will respond to treatment. So a lot of work we're doing at the moment is we're looking at subjects. Individuals haven't developed actually inflammatory arthritis, but they do have these autoantibodies in their blood. So these autoantibodies in their blood suggest that they're at risk of developing RA. So what we have now is we found a signature of a specific immune cell in the blood of these patients, and it's actually, if you measure the signature, rheumatoid patients are up here, healthy controls are down here, but these subjects that actually haven't developed any clinical signs of inflammation, the gene signature is already up. It's not as high as rheumatoid, but it's up in the blood, and those cells haven't tracked to the joint yet. So what we're trying to do is try and intervene at that early stage before disease onset happens. So we found that signature at the moment, and we're utilizing that signature with a lot of computational analysis, mathematics, and doing machine learning to try and predict what patients are doing.
And that group there, if I might just follow on, that group in the middle that haven't developed the clinical signs yet, am I right in thinking that that group of patients, though, they will develop? Do you know that?
Well, we know 60% of them have already. So the group that we're following at the moment, 60% of them have developed, but actually they've developed it a lot. We've delayed the disease onset, and actually the disease has actually been much milder. But again, we still don't know what treatment. So a lot of them, there's the ethics question, as you know. There's the ethics question: do you give a drug to somebody that doesn't have a disease and might never get the disease? Okay. So that's the question. That's that happens in breast cancer as well. So with us at the moment, with those patients, I'm not a clinician, so the clinicians I work with, give the patient the option whether they want to go on to early treatment, and if they don't, when they start to get the first signs of actually early arthritis, then they will go on treatment. And we all know the earlier you treat patients, the better their outcomes are. And I suppose just if you get into kind of more personalized medicine, we do know these autoantibodies in the blood are a sign that they're produced by a particular cell called a B cell. And so for patients with rheumatoid arthritis at the moment in Ireland, generally you're put on methotrexate, and if you fail that, you're put on an anti-TNF. If you fail that, you're put on other treatments. But we know that certain patients should be put on a B-cell treatment from the beginning. So we have some data now, again, in the blood and in the tissue of patients to suggest that actually those patients shouldn't go on methotrexate or anti-TNF initially. They should only go on a rituximab, which is a B-cell target. So we're getting that information because we're able to do much more analysis of the blood and the tissue from patients.
And in terms of what you just said there, in terms of some patients that they will respond to maybe a therapy other than an anti-TNF, which would traditionally be the first biologic. Is that actually happening in practice then? In terms of clinics, would clinicians be identifying that?
Gerry might answer this more than me, but actually I know, so we do biopsy studies. So the way the pattern of these cells, they make a certain pattern in the tissue of patients. So we know when they make that pattern that they're more likely to respond to rituximab. So from the research clinical point of view, we are doing that. So the clinicians in the research are actually, if we see that pattern, Professor Veal, who, as you know, who I work with, actually, who's also married to me, actually, when we see that pattern of B cells, we actually will give those patients rituximab first.
Okay, thank you. Gerry, back to you. So moving on to sort of the patient involvement in research, can you talk to me a little bit about how important it is to involve patients in research to improve outcomes and advances in treatment?
Well, if you're doing translational research, it has to involve the patients. The important thing, I mean, in the NHS, when I was in Sheffield, every patient was really expected to be offered attending the hospital in any specialty, was expected to be offered into research. Now, that happens with cancer studies. Most patients with cancers end up going on to studies, or 40 or 50%. And we try, to do that. It's resource intensive. But the main thing is we've been talking about personalized medicine, is to understand each individual patient as much as you can, and to get a detailed history from them about their, lots of things about their, age, sex, whatever, drug history, comorbidities, smoking, everything, an examination, and record those. And then to link that up with, well, in our case, molecular cellular studies, to see if you can split the patients up dependent on what we've been discussing, antibodies or genetics or whatever. So that's what we're trying to do. So patient-based research is key, and it's about really getting as much information as you can. And the more you can do, the more you can study, the better it is. I about 15 years ago, collected 1,000 patients when I was working in Sheffield, and I got a radiologist to read their X-ray, and we scored the X-rays of the hands and feet. And it was a mark. They got a metric from 0, which everything was normal, to 160, where all 32 joints of the hands and feet were completely destroyed, and there was no cartilage, and there was erosions on both sides, so they had really severe rheumatoid. And then we were able to start to look at things that were associated with severe damage. So if you have both antibodies in the blood, rheumatoid factor and CCP, they had the most severe X-ray damage. And if you had just one of those and you were negative for the other, you had intermediate levels. And if you're doubly negative, it's much less severe, the rheumatoid. And then we went on to look at, in genetic studies. So having patients involved in research and, getting time for research is key. It's interesting. I do a lot of genetics, and in Britain, everyone was skeptical of this. You've maybe heard of the UK Biobank. Have you heard of that? Where they've done this amazing study. They've now recruited 500,000 people to this amazing study. So the patients, they're mostly just, they're not specifically patients or population. That's always a risk that you're going to get people who are interested. But anyway, they've recruited 500,000 people, and they come in, and on day one, they spend 8 hours filling in questionnaires and being examined. And then amazingly now, they've completed the sequencing of all of their genes. And so for some of our studies now, there's Dara O'Leary, who works with myself, and Orla, who's here. She's now using that data to search for genetic variants, and then comparing them to patients we've recruited in Ireland to see if there's a difference, or just to validate what we're doing. But that just shows you how important it is to get detail of patients. These patients are, this has been billions of pounds at the Wellcome Trust, and the NHS has invested, and it's 500,000 people. And, I say it's 8 hours on the first day of recruitment of getting full details of the patients, of their histories, social histories, medical history, family history, et cetera, et cetera. The reason we were all skeptical in this is because, when I see patients, I'll frequently ask about previous medical history, and I'll say, what about hearts? And people say, yeah, I've had a heart problem, I've a heart disease, and you'll ask about it, and it was a bit of chest pain, which is muscular, and it's not really, And so that the gold standard might be to do an arteriogram of the heart and to see if there's a blockage, but you can't do that for 500,000. So you get relatively soft data, and a lot of people were, and I was as well, thinking, that you get soft data on patients, it's not that useful, but it actually has proved to be. If you do it in enough patients, you can get over that weakness to such an extent. And so that's a great example of, it's just not what we were doing 10 years ago. It was just saying we'll collect 1,000 patients with rheumatoid arthritis. It's, what you're doing is getting 1,000 patients, but getting patients involved and getting their full histories and getting their tissue and, biopsies and whatever, bloods, and using that, and it's linking those two. Yeah. And that's proving very productive, not just it's for all conditions.
I mean, in terms of you mentioned the last sort of 10 years or so, I mean, one of the other things that's, in relation to patient research is the actual involvement of patients in research. So in terms of PPI, so people will sometimes hear that word PPI, patient and public involvement in research. Can you tell me a little bit about what that means and how that features in the work you're doing in UCD?
Yeah, so I know Trinity does as well. So we had our PPI conference this week, and just patients and the public, and we had speakers from Ireland and England speaking on, and the patients decide and the public decide what the topic. Last year it was on new treatments, which included prosthetic joints in the thumb, for example, so quite unusually. But this year it was on non-typical involvements, non-typical organ involvement in rheumatic conditions, so eyes, feet, heart, lungs, and that's what the patients and the public asked us to do. So that's one thing we do. And the other thing is when we're writing, when we're doing research now, we all have patient panels, and patients have to really be involved from initiation of the study to, we talk to our patients and the public. We have panels now about any research areas to develop what are the key research areas, and we've done this for Arthritis Ireland, but patients should be involved at the inception of studies and writing grants. So patients are involved in writing grants, particularly lay summaries are very important and very difficult. I used to, AR UK had a 1,500 word lay summary, and I found it the most difficult thing to write in a grant. And we get our patients to help us to write those. And then we've just got HRB grants, and there are patient panels involved in reviewing progress of our grants. Every 3 months, we give them show the data, and they will review the data and give us their opinion on it. And once a year, they meet with all the PIs in the grant. And then at the end, they're important in dissemination. Where do we publish, and how can we get this out in the community, the results of our research. So it's involvement at every level. It's really developed over the last 15 years. So now it's a key thing in doing research. You cannot submit a grant now without actually, and it's not just when you're submitting it, it's a nominal thing. It has to be from time zero.
Okay.
I've got a point to Claire there as well, because Claire has been great from that point of view, and she has actually edited some of my abstracts as well, but also myself and Claire have spoken at some PPI things together. And I think the important, I think the important thing about it also is that, particularly from my side, where I have a lot of young scientists, and they're in the lab. They never meet we're never allowed to meet the patients from a GDPR point of view, but also from the point of view that you probably don't want to meet me because I either want blood or tissue, But in general, they don't get to hear the patient's story, and so sometimes they really need to understand the impact of the disease. And that makes them more passionate. I know Claire, and I keep pointing at you, Claire, but Claire spoke at the Translational Research Meeting in St. Vincent's before Christmas, and it was the two of us in the patient section, and all my lab just loved Claire afterwards because they said it really brought home to them, the impact of the disease, and more so from 20 years ago, because I think they're all in an environment now where we've had all these therapies. So I think it's both ways, and it's really important.
Yeah. And we'll actually have Claire's, after today's event, we will have Claire's talk that she gave earlier this morning available for everybody to watch. So lots of opportunities there to share that as well. Actually, just when you start, you mentioned there about the lab and whatever, that's obviously where a lot of your work takes place. Can you tell us a little bit about that? I mean, in terms of the types of procedures that you undertake in your research in order to progress what you do.
Okay, so I kind of talked about it a little bit earlier on. So actually, I'll just say, a bit like what Gerry said at the beginning, that actually the support to create these two chairs has been phenomenal for Ireland. And actually, I'm the only basic scientist, chair in Ireland in rheumatology. And so when I moved to Trinity, it was basically tumbleweed in the lab. I'd no equipment, I'd no people, I didn't have anything. And so they supported me, but also the funding from Arthritis Ireland also supported some key people in the lab and some key, equipment in the lab. So from that point of view, it really helped me start the lab. But so going back really to what I said 2 questions ago, the 3 areas that we work on is trying to predict disease onset, predict what therapy will respond to, and coming up with new therapeutic strategies. So generally on a Monday, there's a research clinic where I get all my samples from, and patients either come to the research clinic to give bloods, or they undergo a mini arthroscopy. So it's a keyhole surgery where they're awake, it's local anesthetic, and actually I've heard stories from the last 2 weeks where the patients were thrilled because they could actually see, they were awake, they could see actually inside their joint, they could see, maybe they weren't thrilled to see their synovium and their inflammation, But they actually, for themselves, could see actually what's happening inside their joint. And they actually could also see the clinician taking the biopsy from inside their joint. So that's really important from that point of view as well. But so I get biopsies, and there's, it's got quite complicated because, as Gerry's saying, we've got all this technology now. So if I was sitting here 20 years ago, I would have said there's maybe 10 important cell types in the joint. So we've just done the first single cell analysis of RA and PSA in the tissue. And like the initial analysis is looking at, we have 75 different cells interacting, bad cells, interacting with each other in the joint. And they're all producing thousands of proteins, so bad proteins. So I'm amazed sometimes that we've even found the 5 or 6 really good therapies that work because the joint microenvironment is so complicated. So a lot of my work is to try and figure out what's happening in the joint, because that's where all the inflammation is happening. So I take the biopsies, we look at the cells, we try to categorize, is there differences between the patients and the cells that are involved? And some patients would have an awful lot more what we call T cells, and they should go on a drug called abatacept. Some might have a lot more B cells, and they will go on a specific drug that targets B cells. And some might, produce other proteins, and we have those targets as well. So I'm trying to understand what's happening in the joint, but very few clinics, as Gerry will tell you, very few clinics do a biopsy as a routine test for patients, and just economically we couldn't do it. So if we understand the joint, what we really want to do then is to see, can we get a blood correlate, that's something in the blood that correlates with what you're seeing in the joint, and that could be used routinely in the clinic as a biomarker. So essentially we're trying to understand the joint so we can develop better biomarkers to predict disease and response, but also from the joint point of view, I'm also trying to find new therapeutic targets. So that's, in a nutshell,
you've explained it really well. Yeah, no, thank you, because I know it is complicated. So as a non-scientist, I fully understand what you're talking about. Thank you for that. Of course, a lot of the conversation we've had so far is about inflammatory arthritis, and I know that that's where both of your specialisation is. And there's been huge developments, which we've spoken about today already, in the treatment of inflammatory arthritis. That sort of development hasn't happened in relation to osteoarthritis, and I know we couldn't not discuss that today, and we did touch a little bit on it this morning. But obviously, for people here in the room and for people watching, there will be many people with osteoarthritis, and we haven't seen that kind of level of progress. From a research perspective, can you explain a little bit, about why that is and maybe what the future might hold? Geraldine touched on it this morning, but I just wanted to ask you for your opinion on it as well.
I think part of the problem is osteoarthritis is such a heterogeneous condition. I mean, so...
and can you explain what you mean by that just as well?
Yeah, that's right. So I see patterns of osteoarthritis. So knee osteoarthritis is related frequently to a number of things. Injuries to the ligaments or meniscal lesions that happen maybe in sports people. It's being overweight is another issue. Genetics, but it seems that the genetics is different in different joints, which is interesting. The one I find a lot of in the hand, which is very strongly genetic, is hand osteoarthritis. At the base of the thumbs here and here, and then I always ask the patients to take off their shoes, because virtually always it affects the first MTP joints, and then they're at increased risk for here and here. But one thing is it's very different in every patient. It's not like rheumatoid, which is generally the small joints of the hands come on and they're antibody positive, and it. Osteoarthritis, as Geraldine says, also takes ages to develop. And the other thing regarding drug treatment is it takes a long time probably to see an effect from a drug. Because in rheumatoid, the European Medicines Agency, which regulates drugs for rheumatoid, for a new drug to come on the market, they have predefined metrics, so that there has to be a phase 3 study that lasts for 6 months with a new agent, and you have to show a particular that inflammation, swelling, inflammation in the blood goes down, and also that X-rays of the hands and feet are altered, so they're reduced in patients on the drug as opposed to patients just on methotrexate. And so that is but for osteoarthritis to see a significant change in a phase 3 drug study may take 5 years. And that's an awful lot of money for a drug company, although I think the market's there. And that's one of the big things that really limits it. It's such a, you would have to predefine your what type of osteoarthritis, what distribution, which joints, which patients, which risk factors. And then you'd have to do a long-term study, and it's very difficult. And the converse of that is, or well, it's really quite similar in lupus, which is an autoimmune condition that we see as well. That, again, is very heterogeneous. It can affect different organs in different patients. And because I do drug regulatory work, and the regulators find it very difficult to be sure when they look at the results of a phase 3 that the control group and the patient group are actually in any way similar, because it's a really difficult thing to do, whereas rheumatoid is relatively straightforward as a condition for drug studies, because we know, it looks fairly similar. But so a lot of things. So those are things that drive osteoarthritis and the difficulty in finding new treatments. I mean, with the basic thing about osteoarthritis, is it the cartilage? Where's the primary pathology? Is it in the cartilage? Is it in the bone? And frequently it's inflammation as well. Certainly in early osteoarthritis, there's a lot of inflammation, and that's probably the immune system seeing breakdown products of the cartilage, but it may actually be driven initially by inflammation. So, some people we say have inflammatory osteoarthritis, and they come in and their hands are really inflamed and their joints are very, and it's multiple. It's just that the joint distribution is different from rheumatoid. And so it can be very inflammatory. So even the pathological basis, what actually causes it, where the primary abnormality is in osteoarthritis, is debated.
Okay.
Yeah, I think what Gerry said there, one of the real difficulties with it is that actually it presents very late stage. It actually presents when the cartilage is broken down, when your bones are rubbing together, and actually your first idea that your joint is broken down is that you get pain, but the pain comes after the cartilage is gone. So, we're not getting it at an early stage, and it has been really, really difficult. There was one start just back from ACR 2 weeks to the American Congress of Rheumatology, and there was one study there targeting ranked ligand, which is produced by cells that break down bone, and actually it's looking pretty good. It's for a small study, it had efficacy in it. So actually, I think it was phase 2, but actually it was looking pretty good for small joints in the hand. And then there's a bit of a revisit towards anti-IL-1, a slight revisit, because anti-IL-1 we thought was going to be great for rheumatoid, we thought was going to be great for osteo, it didn't work. And then they actually did a huge study, Novartis did a huge study, wasn't it Novartis? Is it?
Amgen, they've taken over.
Huge study in cardiovascular disease, and it had a fantastic result, and it was thousands and thousands of patients. But then they actually did a sub-analysis, a long-term sub-analysis, and they found in a subgroup of OA patients, a specific group, that actually it seemed to work on those patients. They had less joint replacements. So that might be revisited, but it's coming back a bit to this subgroups and personalised medicine and what works for who.
Sure, okay. And I'm going to ask you my last question as well, Ursula, and that's kind of like I'm asking you to take out your crystal ball now. And, look, the question I have for you is, what do you think we might be looking at in the next decade or even in the next 20 years? I'm not sure. I know, research takes a huge amount of time, but in terms of what that might mean or what you would hope it would mean for a patient's prognosis.
Yeah, I mean, ideally, and I think this kind of sums up what we've both talked about, ideally, I show this, sometimes when I give talks, I show the slide at the very end. Some of you might have been at one of my talks before, where there's a patient sitting on, the hospital bed, and they literally have a barcode on their arm. And ideally what we'd like, I wouldn't particularly like to put a barcode on you all, but ideally what we'd like is that we can actually just do a little genetic profile on you or a little blood sample on you, and we would be able to identify a signature that would tell us on the very first day that you will respond to a certain treatment, and that we have treatments for everybody that actually work. And again, the technology we have at the moment is so big and vast that it's basically going to now split into four sets of groups that need to be involved: the patients, the clinicians, the basic scientists, and the computational mathematicians. But I think over the next 10 years, we're going to find out so much about these diseases that I really hope that actually we will find that blood correlate that will actually tell us, and I think tell us as early as possible, pre-disease, that actually patients might be, at risk of developing disease.
Okay.
I don't know whether you have anything to add to that.
It's very hard to predict. It's like asking an economist what GDP is going to be like in 5 years, and you get 10 economists who will give you different. It's very hard to know. But translational research and I think rheumatology, apart from cancer, rheumatology has been the area of medicine that has been really impacted by all of these technologies and drug development, and it still is, and it's still driving a lot of things. Rheumatology is at the forefront. I don't know if it's just our conditions, and particularly rheumatoid, that has allowed people really to dissect, and actually animal models as well, TNF and IL-1 really showed us that they were the things that were driving inflammatory arthritis, and it's proved very, exceptional. It's very hard because things are moving so quickly now to say where we'll be in 5 or 10 years. So we'll wait and see. But
I think you've aptly answered it in terms of where you hope where things are going, and you hope that. I think that's a real gives people that kind of real hope that are watching and listening here today. So thank you very much, both of you.
Thank you.
And we look forward to lots of exciting new developments. From UCD and Trinity, and indeed from all of the other rheumatology research that's happening in Ireland. So it just remains for me to thank you both so much for coming here today. It's been really, really great to have you as part of the panel discussion. Thank you very much.
And thanks Arthritis Ireland for their support over the years. Thank you.
You're very welcome. Thank you.
Yeah.